Hi, everybody. Whew, what a day. Okay, so plan of the day today is to get us started on our sequence analysis, right? So this is a kind of multi-step process, and this is exactly, really, but apart from the organism and gene, what I do whenever I do work of this sort. I haven't really done much of this kind of molecular work, but it's the same principle that applies, right? So essentially what we're going to do is we're going to split this up into two sessions, right? First session, we're going to work on, kind of finding our way around the kind of information sites that are really useful for sequence information, which, because this is a model organism, we have its own website, which is pretty neat. Same as for C. elegans. Um, and also, kind of getting a feel for what this sort of information looks like, right? So that's one part. That's kind of the, I don't know, like the preliminary scouting kind of information. Let's find out some background info. Let's see if we can get some information about function, from uh, what's there already, right? That's essentially what we do first. And I do a lot of this. Actually, one of my students spent a whole semester pulling up this information for, shoot, how many genes? 360 thereabouts. And she got halfway through another list of about 1,000, and she's like, "I'm tired of doing this." And I'm like, "Yeah, fair enough." So that's still to be finished off at some point before publication. But it's exactly what we do, and you can get really interesting information out of that. So for this particular project, we're looking at a large scale expression study, like what genes are differentially expressed either up or down in particular genetic backgrounds, right? Kind of like a fishing expedition. And she actually found, hang on, Adam, let me answer your question in a second. She actually found that a lot of the genes that were being expressed in the intestine of the animals, which was really cool because that's where lipids are made, which is the actual phenotype that we were looking for, right? So even though it might seem a little abstract, it's actually a way of kind of taking lots of information and passing it down to something that we can get a handle on, right? So we can understand it better. Oh, okay. So yeah, we will be going through the different steps for sure. That's a definite. I'm not going to just kind of say, here you go, and like abandon you, like drop you in the middle of Montana with a $10 bill or something, which actually would be kind of nice, at least in the summer. But anyway, so there's a whole series of steps here, right? So this session, we're going to go through that kind of preliminary background work, I guess you'd call it. Next session on Thursday, we're actually going to take that sequence information along with some student generated sequences. And we're actually going to use a software program called Eugene to look at our sequences, right? How do they differ to our reference sequence, right? Where does that difference occur? Really drilling down into the details of the sequence change that results in the yellow phenotype, right? That's the whole point overall. So that also gives us another lab potentially on Tuesday, if we don't need it for lecture catch up, where we can continue that Eugene work. Because that Eugene work is not trivial. And I say that as someone who's gone at Eugene for several semesters until I finally kind of got used to its quirks, right? So that will take a bit of time to work through. So I've actually just uploaded a new version of, well, a slightly improved version of the lab handout to Blackboard. And the reason being is that I've just made a few little tweaks that make it a little bit easier to answer some of the questions, right? And those are were incorporated from the semester just gone, not the summer prior, right? So if you download that anew, there's not a big difference, but there's just a little bit of extra information in some of the for some of the questions. So essentially the way this is set up is it's a very measured walkthrough of all the different steps, right? So you will be learning really useful skills, but you'll be learning them in a very kind of, measured, careful manner, right? Do this and then do that and do so on. And we're doing that in a very deliberate way. And we're also tying that with the questions in the assignment in Blackboard. So for example, if you look at question 1, we're telling you to actually do the first step of finding a gene sequence or finding information about a gene, right? And so the first question is about some of the information that you find. And so every question in the Blackboard assignment is linked to a task that we're asking you to do. So if you go into the actual assignment itself, you'll see that this ties in very much with the step that you're on, right? So don't try and jump ahead too much because it will not necessarily correlate very well, but you'll be kind of led through it in a very kind of careful way. Here's also the PowerPoint on the Sanger sequencing, in case you missed it last time. This is the sequences from the student labs that we'll be working with. This is a zip file, right? So you'll need to download this to your computer somewhere or tablet or whatever and unzip it. You need to extract the files that are in this sequence file. What does that look like? Which one did I use? Good yellow sequences. Right, so when you extract it, it'll look something like this. Right, so these are all of the sequence files that are in that zip file. And I'm guessing that these different letters are initials of the students. You don't need to use all of them. You should use some of them. Right, so you'll need to do that as part of the Eugene section of the assignment, which I think are goals 3, 4, and 5. We'll be doing that on Thursday. And finally, along with the, some of the other videos that you've had the benefit of that Dr. Page made, he also, I'm pretty sure that was him, made some videos to walk you through kind of in person all of the steps that you'll be carrying on today. So not just you're going to have me doing it, but if you don't happen to like the sound of my voice or whatever, you could listen to Dr. Page instead, right? So here's another kind of way of looking at it or doing at it. And, we both cover the same material, we just do it in slightly different ways. So if you go into that document, there'll be some links to, I can't remember if it's a video or videos. It's a little while since I looked at it. That will help you carry out this assignment, right? And so this assignment that you're going to be asked to carry out is a fairly big one because it covers multiple labs. It's a pretty solid piece of work that you're going to be doing. And so there's hopefully plenty of help to help you do it. Even if you don't do it right here and now, you do it in your own time as well. So those are all the resources that are available for you for this assignment. What we cover today will be fairly straightforward. The Eugene one will be harder to do simply because it's going to be using a piece of software you're not familiar with. Right, and it's a really advanced piece of software. This isn't a something that's been made for students to use. This has been made for researchers to use around the world, right? So if I were to do some more of this work and I needed to do some bioinformatics, I would be using this software myself. Right? And it's actually not bad once you get used to its various idiosyncrasies. Okay, does anybody have any questions so far? Okay. All right, so the very first step in any process is really to get at what information is already available, right? You don't have to reinvent the wheel here. You kind of work, I mean, essentially you stand on the shoulders of others, right? So this is all stuff that other people have discovered, and this is all placed into a repository for you to access. And so if you work on flies, which I don't, you would go to Flybase. Actually, I'll show you where I go. This is where I hang out. So this is Wormbase. I find it a little bit easier to use than Flybase simply because I've done, you know, a lot more of it. What was a gene I've been working on recently? Type in a gene, that takes you to that, and this is a relatively, come on. There we go. And there you go. There's me. That's one of the papers I wrote. As you can see, it's not a heavily researched gene, as I contribute like 2 out of 7 or 2 out of the 9 bits of information about it. But that's the kind of way in which you go about finding this information, right? So if you happen to come across this particular gene in, I don't know, mutagenesis or RNAi experiment or whatever, then one of the things that you would read would be the paper that I wrote about cell survival, right? You'd also read, you know, these papers, right, which I have because I was working on that. Questions? Yeah, it's pretty wild, Adam. I mean, like back in the day, we didn't have this, right? So this was really set up by a researcher called Paul Sternberg at Caltech in California because it was such an important thing to have. And it really started developing kind of midway through when I was doing my PhD. So before that, you didn't have this, right? It's astonishing to think that, you know, this is not something that's always been, you know. And if you went and looked at another gene which I've done a lot of work on, which is a growth factor signaling, not only do you get information about it, but if you look at the references, now you can see how heavily studied this gene is. There are 490 individual references, of which 181 of them are research papers. Right, and presumably oh yeah, there's mine. There's one recently from last year. Right, it's an extensively studied gene, right? So there's obviously going to be a lot more information about it and a lot more known about it because more people have been studying it. So these are incredibly useful resources. And there's way more to this than I've ever begun exploring, and I have it on my list to do so. But it's a lot of valuable information here, right? And it takes some time to kind of get used to making the most of it. Come on, get out of it. Move. So the one we're going to use is called Flybase. Flies. Horrible bloody things. Worms with wings. And it's set up differently. So to me, it looks weird. It's like, ooh, that's all kind of ugly, right? Because I'm used to Wormbase. But I'm sure for fly people, it's perfectly beautiful. But essentially, you have all kinds of other information here, similar to, you know, what I've been used to, but more so, right? Because now we have all these other things too, which is crazy. Anyway, so we're going to, not dip our toes too deeply into the water. We're going to use basically the search function, which is similar to the one I showed you for Wormbase. Oh, look at that. It's even got an autofill because that's the only gene I've ever looked for on Flybase, at least recently. And so you type in the gene that you're looking for here. So actually, we could just do something different. Here's another gene, right, which we've worked on, right? This was one of the genes that we looked at for our linkage experiment. So when you go into that, here's white W, which you recognize now, right, from our annotation for the cross, right? It's an ABC G2 class of transporters. I don't know what the hell those things are. That are involved in transporting cyclic GMP, amines, pigments, including drossopterins and omochromes. I don't know what those are. Probably find out. So presumably all of these things, not sure about cyclic GMP, but all of those things are presumably involved in pigmentation of the eye. Right. So as you would expect, mutation of that gene results in flies with white eyes. Right. So this is how you start digging into the information that's already available for a particular gene, right? This is the preliminary step, essentially. And then when you wander through it, you're going to get some information about the gene itself, right? This is where it's at in the genome, right? And so that's the chromosome that it's on, X in this case, because it's X-linked. We already know that. And this is its actual physical position, right? So as a gene, it goes from base 2,790,599 to base 2,700,996, 466. So it is about 4 kilobases long, give or take. So we got all the other kinds of other bells and whistles. We can get like individual bits and pieces of it. We can get the whole sequence. This is what it looks like, right? So this is the chromosome. It's showing you that it's going in that direction, right? So again, this is kind of going back to what we talked about a while ago. Then you can look at function. So these are called Go terms, and that's not like Pokemon Go. It actually stands for gene ontology. And so this is a way of kind of basically you look at a sequence and go, hey, you know what? I've found this little bit of sequence involved in other proteins that are involved in transport stuff. So if you find this little bit of sequence in our unknown protein, there's a possibility it's also involved in transport. That's essentially what a Go term is. Or if I find another little bit which is involved in, I don't know, adding phosphate groups to stuff, like phosphorylation, then we find that in ours, then maybe our protein phosphorylates things, right? And so that's essentially what Go terms mean. And so if you look at it, it's like, Okay, transporter, small molecule binding, transport, seems to be involved in some aspects of nervous system process and behavior, which makes sense, I guess, if you can't see quite right. And it's typically located to the cell membrane, periphery, which makes sense because it's a transporter, right? So you'd expect something that transports things across the membrane to be located in the membrane, right? And that's what all of this is telling us. So we can start building up this picture of what this thing is, where it's at, and what it does, right? Based on, what we're looking at here. And, we can kind of toodle around different things. We've got some different drop downs. Oh shit, that's a lot of stuff. Okay, we're not going to read through that because that'll take all too long. If you wanted to figure out what would be a good allele to work with, or what the commonmost allele is, or even what fly strains have this particular allele, then okay, we look at this. And so these are all resources, essentially, right? So the most commonly used allele is this one, W1118. And it tells you that's a loss of function allele. So if you remember back to when we were talking about different types of mutations, this is a mutation which, this particular allele is a mutation which gets rid of all function. So this might be a truncation. It might be a from a nonsense mutation. It might be a frameshift mutation. It might be a splice acceptor site mutation. It could even be a single amino acid change, a missense mutation. But that amino acid is involved in such an important part of this protein that the protein just doesn't work when it's changed. So it varies. You can't really tell from that. But this will give you the general type of mutation that we get. So we got hypermorph, that basically means only partial loss of function, so on and so forth. So anyway, we're going to do that for yellow. We're going to do that for yellow. Here we go. Slow internet today. Okay, so, we now get like different kinds of yellow. This is, I don't know what the hell yellow C and yellow F are. They're different kind of genes. We want this one. So to get the information, it gives you some basic information here already. So you tell me, what kind of information does just this bit of information here, right? The one with the tick in it. What are we being told here? What kind of information can you glean from that?
The location of the allele.
Yeah, the location of the gene. So the alleles are all going to be in that gene. We'll find out later on where those alleles are if we need to. Right? So yeah, this information here, right? So tells you it's on the X chromosome. Ooh, I might have told you the answer to a question there. Nudge, nudge, wink, wink. And you can also tell, which you already know, right, from your linkage experiment, this is actually fairly close to the white gene, right? Because that was 2 million and something base pairs, right? So this is, a couple of megabases away from the white gene. This is also closer to the end as well, right? So this is kind of way out in the boonies on the end of one of the chromosome arms, which is pretty cool. Now, in terms of how heavily studied this gene is, would you say yellow is more heavily studied than yellow C or yellow F, or do you think it's a much more rarely studied gene? How could you tell? What kinds
The
of information?
references provided.
Yeah, so the number of references, right? So this has 958 references about it, including one which I wrote with a friend of mine, Dr. Page. It's also got 950 different alleles, right? So there are 950 different versions of the yellow gene available somewhere in the world for this one gene. In contrast, these two have 4 and 5, right, which is nice. It's obviously better than 0, but it's still not a lot. Also, if you wanted to get some yellow flies for your very self, there are 53,290 different stocks, right? So that means this gene or some allele of it is found in perhaps on its own or with other combinations of alleles of other genes in over 53,000 different strains of flies, which is astonishing. But it also tells you there's only one transcript. So there's one gene, one mRNA, and from that, one protein. So this is a fairly straightforward, simple gene, relatively speaking. If you look at these, we have multiple transcripts and multiple polypeptides. So these are much more complicated. So we're going to have a deeper look into the yellow gene. So you click on that box, and that will take you essentially to what we looked at for the white gene. So we've got some basic information here, right, about what Y is involved in, right? So you get rid of it. Males can't court as effectively, and the flies are yellow, right? Hence the name of the gene. That's not all of it, right? There's a lot more to it than that, but that's the start of it. So this shows you that yellow gene's going in the other direction, gives you the chromosome, gives you the recombination map, right? So that basically tells you where you'd find it on a map in terms of map distance, like we were talking about for 2 and 3 point crosses. And over here, you can see, well, there's nothing actually known about molecular function. We do have an idea. It's an enzyme, converts dopamine into something else. But you can see here, like it's involved in a whole bunch of different processes.
So whenever we're actually looking at the lab or whatever it asks us, it wants us to only focus on, or do you want us to only focus on the darker complected squares or the lighter ones too?
So this is very much a visual representation. What the lab will actually ask you to do is to actually follow these steps, right? So I'm just showing you all the different sources of information. Go to the ones that you're directed to in the lab, and you'll get all the information there. So it's telling you to go to the summaries lab, right? And that should cover everything that you're going to be asked in the assignment. So one thing that I will be very clear about and is really important is follow the handout closely, because it's been very carefully designed so that if you do, essentially what you're told to do, you'll find all the information that you need to get 100 on that assignment, right? There's no kind of subterfuge or trickiness involved. It's all laid out very kind of clearly in a measured way, so you can find that. So if you go to question 2, right, you will find in there these things. So you just have to go and compare the assignment with these, and you'll find which ones you should tick. So yeah, it's a good question. And I like to show you the whole picture because sometimes things are different, easier to visualize in different ways. But the information you need to refer to is here. And then the next one is talking about alleles, right? So we're going to be looking at another tab. Let's just scooch this one back up so it takes less space. Get out of it. So it's going to look like this to you. You're just going to click on this to expand it. And then we don't care about these other things. We want this stuff, right? So, you know, you could actually put that as 100, put it as all. Actually, I've never done that before. What does that look like? It's gonna be like, yeah. Heck, that isn't that astounding? These are all the different yellow variants. How far do I have to scroll down? Do. All right, my fingers are starting to get tired now. Bloody hell, come on, it's got to be down. Oh, there we go, finally, Jesus. Get to the end, right? So there is, yeah, lots. And to think that all of these exist somewhere in the world, busy flying around their little fly tubes, is kind of astonishing, to be honest. Worms are way better because we just freeze them. We stick them in the -80 and then defrost them when we want them. So I always kind of like look down my nose a little bit at fly people because like, I don't have to maintain all my flies forever and ever. I can just freeze my worms. Anyway, you don't need to worry about that. So these are all sorted by the number of stocks available that contain these particular alleles. So your next question is simply, which is the one with the most stocks? See? And so this is what I mean that, you know, all of these questions are really kind of leading you through the process. Don't consider it like a quiz, because we're not trying to see how much you know about this. We're just checking to make sure you've followed the process that we've laid out for you. So you can find all this information another time if you needed to. Everybody okay so far? All right, so we're going to look at this one because this is the allele that we used or you used in our experiment. So you click on that. Here's another bit. I love these. The people who invented these kind of graphical ways of looking at stuff, I think, are geniuses. Right, so this is our map, right? So here's the X chromosome. This is our particular gene, Y. And this tells you, oh, hang on. The mutation that we're interested in, this allele, is down this end. It's at the beginning of the gene. So whereabouts, what exactly is this? Right, so this is what it looks like. Now we need to go to the nature of the allele. Right, so this gives you general information. It's spontaneous, so it just poof, happened. It's loss of function. Why is it a loss of function? So if we change from, hang on, give you a question here. What's ATG mean in translation?
Isn't it the adenine thymine guanine codon that's attached to the tRNA? If it were, in this case.
So it's not the anticodon. We're talking about the codon in mRNA. What amino acid, can you remember what amino acid the AUG codes for? And where would you find that amino acid? Encodes methionine, correct? And it's the start codon. Awesome, exactly. So I think you're on the same kind of track there, Adam. So the ATG or AUG in RNA is the start codon. So this is telling us why one allele is a change from ATG to CTG. What happens to the start codon? Do we now have a start codon for our yellow gene? Because there's usually only one, or at least it's the one that at the very beginning. So if ATG is changed to CTG, can that now act as a start codon? If you're thinking about translation initiation. Absolutely no. Thank you for answering though. So if you don't have an AUG, you don't have a start codon. So if the whole initiation of translation relies on there being a start codon, do you think the yellow gene will be translated if it has this mutation that turns it to essentially a CUG? So it can be transcribed, right? It doesn't affect transcription at all, but can it be translated? If you don't have a start codon. Adam's like, I'm not going to answer now. Got a 50% chance of getting it right, right? Anybody willing to take the chance? Don't forget, this is a loss of function allele, i.e. the protein doesn't work. Or there is no functional protein. Yes, is he? If you change that base, if you change any base in the start codon, doesn't matter which one it is, first, second, or third. If you change any base, it no longer acts as a start codon. So if you don't have a start codon, you don't have a protein. If you don't have a protein, you don't have whatever function that protein provides. So this change here that I've highlighted, one single base, an A to a C, so we have CTG instead of ATG, that is sufficient to completely get rid of all function of the yellow gene. Complete loss of function. So that should help you answer questions 4 and 5. Does anybody have any questions? The NCBI section, Adam? Okay, cool. We can work through that. Okay, so essentially you can get sequence data from here, right? You don't have to go to the NCBI if you don't want to, but the NCBI is a central repository, National Center for Biotechnology, Bioinformatics Information, something like that. I can't remember. So we're going to go to the horse's mouth, so to speak, and get it from there. Or not, as the case may be. The hell? Oh, that's why. Oops. All right, this is where we're supposed to go to. Okay, so this is natural sense, biotechnology information. I was right the first time around. So this is even more kind of intimidating, right? Because this is not cutesy and nice and everything. It's just like raw stuff. These are you know, all the things that you would want to use to do work on sequences, for example. I've used this one a lot. That's kind of why I come here the most. So when you want to search, you have to be somewhat specific. So if we just put in yellow, if I can spell it right, this is what comes back. We've got 5,000 books, you know, 68,000 whatever's in PubMed, which is like the journal repository, 197,000 genes. We're like, oh my God. If one were Janice in Friends, easily my least favorite character. So, don't want yellow fever virus, thank you. You could try narrowing it down a little bit, which makes it a little bit better, but not a whole bunch. Or you could just copy this, including the quotation marks, I'm going to tell you, Dr. Page is an utter whiz at this stuff. He makes me look like a rank amateur. Oh, great. Now you get to get zeros on everything. That's not very useful now, is it, Dr. Page?
Whenever I tried it, I just did yellow and Drosophila organism and I didn't put the quotations. But I don't know if I did it right.
Oh, okay. Maybe I'm being too clever for my own good, huh? Okay, scratch the bunny ears. There you go, that's a little bit more manageable. So, does it tell you what to do next? Let's go to the gene. All right, now we're getting somewhere. Okay, so we've got a whole bunch of crazy stuff, right, which you just don't want to get involved in, so don't worry about that. This one, though, is what we want. So you can do some checks. You can go, okay, does it say yellow? Yeah, says yellow. Am I with the right species? Yes, says Drosophila melanogaster, not whatever these things are, Drosophila virilis. It's presumably a virile fly. Wouldn't want one of those around. They produce enough without any extra virility. And it tells us on chromosome X, right? So this is kind of a nice, yeah, this must be what we're looking at. And so now there's a little clicky bit there under yellow. Click on that. And that will take you to all of the sequence information available for the yellow gene, right? Again, it's in D. melanogaster, tells you here, and chromosome X near the end, and so on and so forth, right? So there's an immense amount of information on here, right? Absolutely huge amount, right? And so we're going to go through this very carefully and very steadily, right? And I'm actually going to refer back to the handout because I want to make sure I'm going to take you through it in the correct way and I don't kind of cut any corners, right? So, there you go. It says D. melanogaster. It's really important. Now we're going to go down to a section that says genomic regions, transcripts, and products. So we're going to go down. So this is the area that we want. Let's just wait for Robert to come back. Oh, there he is. So this is kind of like a amped up, supercharged version of what we were looking at in those other web pages, Wormbase and Flybase. So this is a whole bunch of information on all kinds of stuff. But what we care about is this one here. Okay, and then you get this like magic pop-up thing. Right, so I'm going to get off of that for a second. And I'm going to explain to you what you're seeing here. Right, so this line up here is the chromosome. Now, what are you going to do now? Shit, I don't know. And this, you have to be careful what you hover your cursor over, right? Because there's all kinds of stuff that pops up. So this is a diagrammatic representation of the yellow gene. You see it's got a little Y up there. And it pops up again, right? And so that tells you, okay, we've got the yellow gene, tells you what the length is, right? This is important information we'll get back to in a little bit. And a bunch of other, little bits and pieces, right? Don't worry about that too much. Now, this is showing you that, remember that diagram that I made of a classic, or not I, but Dr. Page made of a classic eukaryotic gene structure? What different things are we going to have in a eukaryotic gene? What features in terms of things? What are we going to have? What do eukaryotes have that bacteria don't in terms of genes? They're the things in between the other things.
Nuclear membranes.
Oh yeah, that's true. Yeah, so as organisms, yes, but we're talking about gene structure. So good answer, not the context we're looking for. Do eukaryotic genes have one continuous exon that encodes the protein? Eukaryotic genes continuous. So the protein matches perfectly with the DNA.
Yes, because the DNA is considered linear compared to the prokaryotes, which isn't. So hence it lines up perfectly.
Kind of, I think you're getting confused with linear versus circular chromosomes though. I think that's where that's coming from. And Robert, yes, they do have operons and eukaryotes tend not to. But let's think about just a single gene, right? So If you were to transcribe the mRNA from a gene in a eukaryote, right, without doing anything to it, would that mRNA be able to be translated into a protein once it's exported out of the nucleus? No processing whatsoever. Would that mRNA be able to be translated properly in a eukaryote without any processing? In prokaryotes, yes, for sure. In eukaryotes, no. Why not? What has to be done in eukaryotes? What has to be removed? What's the word that I'm looking for? Begins with in. Yay, thank you, Maria. Introns. Right, so too slow, Robert. Yeah, that chat box is brutal. So every eukaryote gene has at least one intron, right? It's really, really important. And that intron is this bit here. So the straight line, which doesn't have like a box associated with it, that is the intron of the yellow gene. It's a pretty big one too. Okay, so if you have introns, what else do you have? Shares a similar root, also ending with on. What's the bit that codes for protein?
Exons or exons, something like that.
Exons, you're right, Adam, exactly. And so those exons are the dark green parts of this diagram. So again, this is, what we showed you in class. It's exactly what you're seeing here. What about between the start of transcription and the start of translation, kind of 5' to the first exon? What region is that called? It's 5' and it's not translated. So what do we call it?
I would say the 5 cap, but I'm pretty sure that's wrong.
Well, there is a 5' cap. You're actually absolutely correct, but that's not a sequence feature. It's something that's added to the very, very beginning of the transcript. So you're right, it is there. Maria, the promoter is in the DNA, so that's not in the transcript. So the promoter would be at the beginning, kind of, where those letters are, NM whatever. The promoter would be there and to the left of that, right? So you're correct, there would be a promoter, but that wouldn't be part of the diagram we're seeing here. That would be part of the rest of the DNA. So what do we have? What is attached? What is the 5' cap attached to? What does the eukaryotic ribosome, the small subunit, assemble on before it wanders down the transcript until it finds the start codon? Yeah, exactly. The 5' UTR. Thank you, Robert. And it's in the name. It's the 5' untranslated region. So it's part of the transcript that is not translated. And so we have a 5' UTR here. That's that pale green box. And at the other end, which to kind of riff off of what Adam said, there would be the poly A tail added to, right? So we've got 5' cap and poly A tail on either end. That's the 3' UTR. So we have all of the normal features of a eukaryotic gene. We have our 5' and 3' UTRs, we have exons, we have introns. or intron, right? Other eukaryotic genes, you may have more than one intron. Sometimes you can have several. This one, we just have one. Okay, so if you want to know where those are, hold that over and it will tell you a bunch of information about some of those features. So let's just double check. What are we being asked? Okay, so what proportion of this span of this gene corresponds to codon sequence? Well, you hover this over here and it will tell you the span. And it will tell you the codon sequence length. So the answer is simply 1 divided by the other. If you want percent, you times that by 100. Is this helping, Adam? Kind of taking you through step by step?
Yeah, it was. And then also there was like more parts to where it's like, okay, how many this and that are there? And it kind of threw me off. And also another thing that took me by surprise was where we had to enter in some numbers. So I was trying to
Okay.
look for those on this and I couldn't find them.
Oh, I'm not going to tell you the answer, Robert. What do you think I am? Some kind of amateur professor here? Look in the Blackboard assignment. You'll only find one correct answer. Honestly. Yeah. Kids these days, I just don't know. So, well, that depends on what you divide it by what you're dividing it with, doesn't it? But yeah, Adam, I'll give you some tips about that. So that information isn't super easy to get from here, but it's an absolute piece of cake to get from Eugene. So let's just not worry about that too much for now, and we'll go through the steps of getting the sequence data, and then I'll show you how to get that information, right? It's kind of like something I agree with you. It's not super easy, but when you use Eugene to look at it, it's really piece of cake. It's all written out for you. Okay, so just kind of checking ahead. All right. Where's the? Oh, here we go. So here is the information, right? And yeah, and the thing that's kind of difficult is that you have to hover your cursor exactly over the right thing for it to show the information. So I'll show you here, but then we'll also have a look at it when we look at it in Eugene, and you'll see. It's fairly obvious once you kind of know what you're looking for. So if the top part here, this bit, is the whole gene, then so this is the DNA essentially, but the DNA that corresponds to the gene and not necessarily to the regulatory regions, even though they're actually part of the gene too. This is kind of like the defined part of the gene, I guess one would say. So what do you think the kind of weird shade of purple corresponds to? Right, if it doesn't include this bit in the middle, get out of it. Right, but it does include these purplish looking blocks at the end. What part of the steps of gene expression do you think that purple bit represents? If the green part represents DNA, what's next? What comes after DNA in gene expression? Protein? mRNA. Yay, exactly. Right. So this is exon 1, this is exon 2, and they're stuck together. So this should give you, there we go. Well, it's actually mRNA because this straight line in between doesn't exist, right? So it's, even though it's kind of laid out the way it is, so you can compare each bit with another bit of the gene, which makes it a little confusing because it looks like it hasn't been spliced, but it has. Okay, so this is only 203.38 nucleotides long, so something's been removed. Now I'm hovering over the first part, and that gives you exon 1 out of 2, because there are 2 exons. And if you hover over here, this will give you exon 2. And again, I understand totally where you're coming from, Adam. It's a little tricky to figure out numbers and stuff from this. Okay, but we're just going to work with this for a little bit longer because it's kind of a nice visualization, essentially. Okay, so if that purple bit is mRNA, what's the red part? Bless you. And a small gerbil squeaking over in the corner. My youngest daughter has the very cutest nieces. Although she doesn't like hearing that very often. She doesn't like me sharing that online either, but oh well. Okay, so what is the red bit? It's missing, hey, get out of it. It's missing the bit at the beginning and the bit at the end, the untranslated regions. So what is the red stuff? What does the red represent? It's not DNA. It's not mRNA. It's nope, it's not anticodons. Those are in tRNA. It's not tRNA. Those are encoded by other genes. Sorry. It's protein. So the start codon is about here. Stop codon is about here. So again, this is just a way of looking at it, right? And once you see it, it's like those kind of dot patterns and stuff. Then it's like, oh, right, that's what it is. You know, it becomes a little bit more obvious. That's why we're taking you through these steps bit by bit. So what question are you being asked? Okay. Okay, so question 7. So I'm just going to my memory what the questions are. I think, yeah, there you go, Robert. That's the sequence hypothesis. What is question 7? Actually, let's just go and have a look. Here's question 7. Come on. And you get, oh, I forgot to say, you get 3 attempts at this test, assignment, whatever you want to call it. Okay, so you just got to look at the picture. And do these apply? Are these true statements? The first exon in yellow is the longest exon in the gene. exon 1, according to this. You see here. Exon 2. Ah, quit it. Come on. So is exon 1 longer than exon 2? You pick which one you think is right. Yellow does not have a 3' UTR. Is that correct? Go back to here. Well, I'm not going to confirm that, Adam. Robert's already tried that gambit once already. Think I'm going to fall for it twice? Honestly. Okay, so here's the 5' UTR, and here's the 3' UTR. Oops, wrong button. Does it have a 5' UTR? How many exons does it have? Does it have only one intron, or does it have multiple introns? So if you use this information, you should be able to answer. If I can get back to it. Come on. Go away. Here we go. You should be able to answer this question without any problems. Now this is probably actually, you know what? You don't even have to look at anything for this. All you need to know is what is the basic structure of a eukaryotic gene. Sorry, my neck's beginning to ache a little bit. What comes first in a eukaryotic gene? After the promoter, we're talking about like the actual part of the gene which is transcribed. What comes first? Maybe. Yes, it does, right? So if the 5' UTR is first, I tell you what, I'll give you the first couple, right? And then you can figure out the rest from there. If the 5' UTR is first, it's going to be from the very first base, right? Does that make sense? So this can only be A. What comes after the 5' UTR? Is it going to be the 3' UTR? No. You're crazy, Professor Crook. It's going to be exon 1 because that's the first exon, right? So what's number 172 onwards? That's going to be E. Right. And so you just work through. You don't even need to know what the yellow gene looks like. You can just work it out based on what you know about eukaryotic gene structure. But when we look at Eugene, I'll show you how you can also work it out using the information in Eugene too. It just gives you a bit more information in a more accessible way. So there's two different ways you can do it. A priori, based on no prior knowledge of the yellow gene, only what you know about eukaryotic gene structure, or you can actually just use the numbers from the diagram in Eugene. Either way works, you'll get exactly the same answer. And I believe the final thing is getting the actual gene sequence. Right, so there's no question associated with this. We're going to use this sequence in UGene to, oh no, we're actually going to use UGene. I'm sorry, I was just reading ahead. So let's go back to that page, and we're going to go to, again, follow these instructions. Right. We're going to go to genomic regions and transcripts, which is, where'd that be at? Up here. There are lots of different ways you can get to this information, but let's follow this, the instructions. And I'm pretty sure it's go to reference sequence details. Yep. So you click on this hyperlink up here. And that will take you to this page down here. GenBank link under the header that reads NC. There we go. So this is the bit of the page you need to look at, because underneath here is mRNA and proteins, and we don't want to use that. Not right now anyway. And you're going to need to click on this link, GenBank. And so now what it gives you is like a whole bunch of information. Dum, dum, dum. Dum, dum, dum. Actually, it gives you the mRNA and the exon numbers here too, if you're willing to dig into it. Right, so that's another way that you can get at that 5' UTR, exon 1, intron, exon 2 and 3' UTR stuff. But we're not going to worry about that. The actual sequence is here. This is what it looks like. 4,000 and a bit bases long. Now, right up at the top, which should also be in the instructions, you want to click on the send to. So click on this. And you want the complete record because there's loads of useful information in there. And you want to save it to file in this format. Pretty sure it's that format. Yep. And that will hopefully do something. We don't want to save it in there. So let's save it. Teaching, genetics, lab. Where are we at? Eugene. And save it whatever you feel appropriate. So this is the yellow 1 sequence. Click save. And it will save it down there. Okay, so whatever folder you want to call the folder, wherever you want to save it, but save it to your computer because you'll need to use it. And it's convenient just to make a folder, even if you delete it later, where you put all these sequences, just so it's easier to find them, right? Because you'll be doing a lot of opening and closing stuff. Okay, so now we're going to go and use UGene. This is something you're going to need to download from the internet, right? It takes a while, so don't hang about. Well, just do it the night before you need it, whatever, it doesn't really matter. But it takes a little while to load it because it's a big package. Okay, let's put Mr. Robert over there. So I'm going to just get rid of this because we don't need that. And I'm going to open that yellow sequence. Okay, so called it yellow 1 sequence. So just click on it. And now it'll open up this. So again, Adam, this is another way of kind of visualizing and gaining access to that information, right? So here's our yellow gene in that kind of weird greenish color. Not even sure what you'd call that. The bar in the middle. This is our, hang on down here. This is our mRNA. So this is going to be exon 1 and exon 2. And this is the coding sequence. So this is what gets translated into protein. Let me just double check what, get out of it. What's the next question about? Oh, we've already answered that. Oh shoot, we've already answered that question. Super cool. Kind of getting ahead of myself here. So if you look at all this information, let me just double check. Make sure I'm not missing anything. Yeah, he's questioning. Sorry, I was jumping ahead. That was my fault. Now, when you look at, get out of it. Have to move things around. Tiny laptop problem. Now, when you click on these give you the values. So if you look down here, this is the start and end and beginning and end of exon 1 and 2. Or you can just hover over. So again, this gives you that same information, just kind of different place. So this is, I'm sorry, this isn't exon 1 and 2. This is 5' UTR exon 1, exon 2 and 3' UTR. Exon 1 is the coding sequence at the beginning and the coding sequence over here. So if you just hover over it, it will give you the actual, god dang it. It's hard to show online because I can't move my cursor. You see, like where I'm pointing at my screen, the first bit of information where it says location, the bit in parentheses after that, those are the bases of the beginning, start and end of exon 1 and 2. Sorry for the confusion earlier. I was looking at the mRNA, which has the 5' and 3' UTR. And just for giggles, while we're here, let's kind of get rid of that for a little bit. Kind of invigorate this. No, make it bigger. So this here is the start of exon 2. We're going to go way up. Okay, so give you a hint. This is actually the sequence from the mutant, yellow 1. I don't know why NCBI has it as the reference sequence. This is not the wild type sequence. This is the mutant sequence, the yellow 1 sequence. And so here you can see that mutated start codon. The first codon of exon 1 is CTG. It should be ATG. And so this kind of light bluish color is exon 1. And now this is actually kind of neat. Remember, we were talking about splicing, and the splice acceptor site is always a GT-AG, GT at the beginning of the intron and AG at the end. What do we have? We have a GT. And if we scroll all the way down to the beginning of exon 2, which is way down here somewhere. Here we go. We have an AG at the end of the intron, right? So it shows you how conserved, even that this gene, randomly as it is, has exactly the same splice acceptor site as we were talking about in class. And the PCR product that we're going to be working with basically corresponds more or less with a bit of the 5' UTR, exon 1, and I think a bit of intron as well. So play around with this before Thursday so that you're a little bit familiar with it, and then we'll use it, to look at those sequences and put things together. But that's enough for today, I think. Cool. Anybody got any questions? All right. Well, thank you again. This LabVIEW shouldn't be Thursday. Let me just double check. This should be due next week, so that you have time to. Come on, where is the? Here we are. Yeah, there you go. Sorry, my brain was all kinds of befuddled for a moment, Adam. It's next Thursday. So not this Thursday, Thursday following. And the reason for that is it gives us time to go through it on this Thursday, maybe on Tuesday next week as well. And then it gives you time to finish everything up before the deadline. All right. Thank you very much, everybody. Yeah, me too, Marcus. I'm going to make myself another cup of coffee. And any questions about any of this stuff, either ask me by email or we'll go over them on Thursday. Cool. All right. Have a fun rest of your day. I'll see you all on Thursday.